Hi everyone, welcome back. My name is Laura.
And I'm Adam, and we're your hosts for the CARRM Profile Podcast.
Today we're here with Yina Tian
Hello, everyone.
and Nouran Albana,
Hi, everyone.
who are both master students.
Okay, so let's get into it. Can you tell us a bit about yourself, guys, and your background before entering the field of regenerative medicine?
Hi, everyone. Really excited to be here. Thank you for having us, Laura and Adam.
Thank you. for coming.
So I'm Nouran, and I am currently in my first year master's in the Department of Physiology and Pharmacology at Western. And I did my undergrad here at Western also, and I was in the IMS Specialization in Medical Science program. And I'm really excited to be starting my research journey. And yeah, I guess we can dive into the more details after your questions.
And I'm Yina, and it's great to be here too. So I did my undergrad also at Western in physiology and pharmacology, and I did an honors specialization within MedSci. And that's actually how I got into my current master's position. So I stayed in the same lab that I did my undergrad thesis in, and we're both in the same lab, so we're in the Hess lab, and that is within the physiology and pharmacology department at Schulich.
Very nice. Thank you for the introduction, guys. Okay, yeah.
So what sparked your interest in regenerative medicine and why did you decide to pursue it at the master's level?
Okay, so I was always interested in research, and I think I decided that I wanted to pursue research at the end of my third year in undergrad, after I decided that medicine wasn't for me. And at that point, I really was interested in immunology for the most part, and that's what I had been planning to pursue after undergrad. However, my current PI gave a guest lecture in one of my IMS classes, and he presented his research on diabetes and regenerative medicine and the use of stem cells as a therapeutic for diabetes. And it really grabbed my attention, and I really loved his work. So I spoke to him about taking his class in the next semester, and that's how I ended up being exposed to the field of regenerative medicine, and I only fell more in love with it. And then I talked to him about joining his lab as a master's student, and that's how it worked out for me.
Could I ask you a question? So
Yeah.
you said that you got interested in you had that presentation, you saw the thing about the diabetes and the regenerative medicine. Was it your interest in the topic, or it was
Yeah.
that made you what did you become
Yeah.
interested in the topic, or did it make you interested in doing research about the topic?
Oh, very good question.
Yeah.
Kind of both, because I hadn't considered the topic of diabetes before. I was always looking into immunology and autoimmune diseases, but he works a lot on type 1 diabetes, which is an autoimmune disease. So when he presented his work on that, I was like, wow, this is it is immunology, but it's also incorporating regenerative strategies and using stem cells to combat the autoimmunity that goes on in type 1 diabetes. And that really what sparked my specific interest in that.
So for any of the listeners that don't know about diabetes 1, could you explain a bit about the disease? So
Oh, sure.
Yeah.
So type 1 diabetes is the autoimmune destruction of your insulin-producing cells in the pancreas. So you need insulin to regulate your blood glucose levels after every meal, and your body can't do it without insulin. When your pancreas doesn't produce insulin anymore, that's when you have either type 2 or type 1 diabetes, and the difference between the two is in the pathology, or in how it starts, the cause of it. And in type 1 diabetes, your immune system actually starts accidentally targeting your beta cells that produce insulin. And so it kills them off, so your beta cells cannot produce insulin anymore. And so you don't have insulin that is being produced in your body and regulating your blood glucose levels. So then the frontline treatment is exogenous insulin treatment, which is administered by injection usually. And I think we're all familiar with that.
And also, just to clarify, so type 2 diabetes isn't when your pancreas doesn't produce insulin, but it's when your body can't use the insulin that it produces.
Okay. So I'm assuming that when you're mentioning the treatment for the type 1 diabetes as being exogenous, that means that you guys are looking for like more optimal treatments for that disease.
Exactly.
And what would that be? If you could explain
Exactly.
that?
So exogenous insulin treatment, although it can manage blood glucose levels, it doesn't address the actual root of the problem, which is the autoimmune destruction and the attack. And we call it insulitis, which means like the inflammation inside your pancreas or the islets where the beta cells are, and the immune cells infiltrate that area and kill off the beta cells. So solving that problem, stopping the autoimmunity, would solve type 1 diabetes.
Because then the cells would come back and they could produce insulin.
Because then, yes, like they wouldn't be killed off.
Okay.
So what we try to do in the lab is we try to suppress that autoimmunity and drive the regeneration of these islets. And islets are in their nature, they're not very regenerative. So we try to push the regeneration of them with using stem cells. Islets is beta islets. So they are the area where beta cells exist in the pancreas, because the pancreas is exocrine and endocrine. So it has both functions. So the islets is where the endocrine portion is.
Okay, I see.
Yes.
Sorry, I just have a question as well. So the guest lecture that you had that like kind of got you sparked into it, was it like during a regular class lecture when they have to and talk about it?
So it was one of our IMS specific classes, and those classes, I think it's called Selected Topics in Health. I think that's what the class is called. And the whole class was about diabetes. So every week we would have different lecturers targeting a specific aspect of diabetes. So it went from like bioengineering to stem cells, where I met Dr. Hess, and or like all kinds of different like perspectives on diabetes, how to solve the issue, how to deal with the like epidemic, and yeah, that's so they bring out guest lectures.
I do think they've changed the topic since.
Yes.
So I think it's
cancer
anymore.
now, but it's usually like for 5 years they would do the same topic. So I guess I got lucky. I was on the last year where they were doing diabetes. Yeah.
Just in case anyone wanted to
Yes.
do this.
That's good though that you got it like the right time.
It was. I was definitely lucky. I was definitely lucky in a lot of aspects.
So, Yina, what about you and your research? You want to talk a bit about that, or?
Yeah, so my background and how I got into this field a little bit. So after my third year of undergrad, I was actually volunteering in a lab, and that was because it was pretty much the only research position that I could find on short notice because my plans had changed. So I was volunteering in a lab in Toronto that focused on stem cells and their use in the intestines in babies, and that kind of sparked my interest in the use of stem cells. It was a different field or different disease than what I'm studying now, but through that lab, I heard of my current PI. They were talking about him one day, and I was like, oh, I recognize his name because I saw him in our list of faculty. And coincidentally, that was the year that I was applying for my undergrad thesis project, and he was on the list of candidates. So I looked into more of his research, and I thought it was really cool, and I applied for it, and I've been in his lab ever since.
Wow.
Very nice. Okay. So what are your research in particular, like, because you said that your projects kind of differ slightly. Could you maybe go in more depth about what your thing is about?
Yeah, so I don't focus much on the autoimmune portion like Nouran does. My project, I kind of switched from undergrad to my master's project. In undergrad, I was also on the regeneration side and looking at factors to help induce this regeneration or the beta cell regeneration.
Okay.
But now I'm looking a little bit more into replacement strategies. So as Nouran said, in diabetes, in type 1 diabetes, your beta cells are killed off. And innately, they don't really repopulate too much. So instead, I'm looking into strategies to replace these cells. And right now, there are protocols that use donor cells from people who have recently passed away, so cadaveric islets. And they transplant these into recipients who don't have beta cells or don't have enough beta cells. However, the problem with this is that there are not enough donors compared to the people who have diabetes.
And who need it.
Yeah. So this is there's a really big discrepancy in these numbers. And so one thing that the field has been looking into is differentiating or creating these beta cells from stem cells. So getting a little bit more into the nature of stem cells. So these are cells that can repopulate, so they can self-replicate, and they can also differentiate into different cell types depending on the type of stem cell that they are. So the stem cells that I work with are from a human embryonic origin, and I'm trying to kind of push them more into beta-like cells and cells that secrete insulin. And this is very preliminary research, so it's preclinical. So it's not ready to be implemented into humans, at least the work that I'm doing. But that is the goal for the future.
So just for me to understand, so your goal is like the replacement therapy. So like these beta cells in the pancreas are dead, and then you want to replace them with like these cells coming from cadavers or from humans that have just passed. And your research, like, so just for me to understand, the difference is that you're targeting the autoimmune process, or like what? Maybe just so you can differentiate.
Just to clarify, the cells that I use are not from cadaveric origin.
Oh, sorry.
Yeah, no worries. They're from embryonic. Okay, yeah.
But the discrepancy is like, what is the discrepancy then?
Yeah,
That's
so
what currently exists.
Currently, what a lot of people are using are islets from cadaveric sources, but there's not enough for it for people who need it, so we need to like create them instead of harvesting them.
I feel you. So it's like so now you're using from the embryonic origin like those cells to create more beta islet cells. Like you're targeting them to differentiate to form them.
Exactly.
Yes. Because that solves the discrepancy issue, because you can get them.
Right. So we're thinking that these cells can create an endless source of these beta-like cells, and there's no cap to how many we can produce. Yeah. And so if successful, we would be able to solve that discrepancy between the number of patients who need it and the number of beta cells or islets that are available for transplantation.
Because then you just have like an infinite practical
Exactly,
source of
yes,
cells.
Correct, yeah.
So if I want to just ask one question, like I know it's not related to your research, but like I'm just genuinely curious. So if most people are using the cells coming from cadavers, like I guess how could I word myself? Like, why is it that there's like such a shortage? Or maybe even like, can't they just take those cells out of the people's bodies and then like make them divide to produce more? Like, why can't they do that?
Right. So beta cells are adult cells. Yeah. So they don't really replicate and proliferate too much. So because of that, we're not able to kind of just make them repopulate indefinitely.
It's a transplantation procedure. So it's like kind of a heart transplant. You can't take a heart and just make it repopulate and have infinite hearts.
Yeah.
Okay.
You know what I mean?
It's also because like the stem cells are what give rise to those adult cells that are functioning. So just going back to your previously, so your thing was like targeting the autoimmune process?
So I guess we haven't really talked about the specific type of stem cells that we use in the lab to push this regeneration of the beta cells in the pancreas. So I guess I'll talk a little bit about them. So they are called mesenchymal stem cells or multipotent stromal cells.
Complex name.
Or MSCs. I'm going to call them MSCs from now on. But they are non-hematopoietic precursors of fibroblasts that come from the bone marrow. I'll explain.
Okay, yeah, you got it.
Should have seen their faces.
That's even a little hard for me.
So, yeah, it took me a while to memorize that too. They basically come from the bone marrow, and they differentiate into adipose tissue, so fat tissue, osteocytes, so bone tissue, or what's the third one? Chondrocytes, right?
But they're not only found in the bone marrow. They're found
the ones we use, we get from the bone marrow, but they can also be found in umbilical cord blood or even adult adipose tissue. Yes. So what we do in the lab is we use bone marrow derived MSCs and we culture them. And so those MSCs have both regenerative and immunomodulatory secreted factors. So the proteins and the factors that they make and they secrete out of the cells, of the cell, they have regenerative capacities and abilities, as well as abilities to dampen the immune system by inhibiting lineages, by inhibiting certain processes that are pro-inflammatory or pro-autoimmunity.
So you're saying
Yeah.
the proteins that these cells
Yes.
secrete are going to be stopping the immune cells from doing their job.
Ideally, or
Ideally.
that's what we're trying to
And what was
test
the other
out.
function that you said?
Regeneration.
Regeneration,
So we
okay.
have shown in the lab
Yes.
that they are able to, once these secreted proteins are transplanted into mice that have had their beta cells killed off by a drug,
Yeah.
they are able to, so these mice turn hyperglycemic, or their glucose levels go
Wow,
up,
yeah,
right? So
okay.
that's how we know they have diabetes now.
Yeah.
So when we transplanted these secreted factors, their blood glucose levels went down.
Okay.
So we have shown that they are able to regenerate or have some capacity to regenerate the beta cells, but we haven't shown that they can suppress the autoimmunity, and that's where my project comes in.
Okay, so these
Yes.
proteins ideally would be like it's like a cocktail of proteins, I'm assuming, would be like a 2-in-1, regenerating the cells and then like killing the immune cells off.
That
Okay.
that is an overview.
And
yes.
your project is trying to make sure, or trying to find a way for the proteins to knock out the autoimmunity side of stuff.
Maybe not knock out, but
Not-
definitely lower and dampen.
Yeah,
the
dampen
autoimmunity.
it. Yes.
Would it be
yes.
bad if it knocked out completely?
Well, you don't want to inhibit all your immune cells.
No,
because
because that would be bad for
so you
you.
get really sick and stuff.
Yeah, yeah.
And out of curiosity, is there a specific reason why you use the ones in the bone marrow rather than the other sources?
Yes, so we have, like, our lab has tried also, maybe not adipose tissue. Have they tried adipose tissue? I know we did umbilical cord
Our
blood.
lab has used cells from different sources. It's just right now our collaborator, who works at the hospital in London, what he does is he tests patients in the area, their siblings, to see if they're a match for their siblings' disease. So I'm not exactly sure what disease he works with, but it's a clinician who has extra samples, and the samples he essentially gives them to us.
I see. Okay.
Yes. Yes. So those patients are undergoing surgeries to for bone marrow transplantations for their siblings that are sick, that need the bone marrow
understand. So they give
transplantation.
themselves.
So we get permission, obviously, and consent from those patients to get an extra sample of their bone marrow to give to research, and that's how it comes to us.
I see.
Okay.
So it's just kind of like out of convenience a bit.
I know there is also studies done comparing MSCs coming from umbilical cord blood and bone marrow. They are comparable, so like their regenerative capacities are similar. So we just use the bone marrow.
It's easier for us.
It's easier.
To get,
Also, umbilical cord blood MSCs are harder to grow and maintain.
It's
in
also
culture.
commercially available, which is one avenue that we are potentially looking into. So there are companies that, like biotech companies that do sell these types of cells,
Okay.
that are already ready to use, essentially.
Let's say like I wanted to go to one of these companies and buy like, I don't know, like let's
You
say like
can't.
1 kilo. Okay, let's say I
You
was
can't.
in a position where I could buy them, yeah? Like say I wanted to buy like a kilogram of these cells, like how much would that cost? Like
I don't know if a kilogram exists.
It does
No,
this-
I'm joking.
Okay,
They're
okay.
about $1,000 for one vial, I think.
Yeah.
Wow. And a vial is like if for
It's
you,
1
like,
mil, I think,
1
if it's
mil.
if it's the
I think it's
same.
more.
So is that like
Is
a,
it
like,
more?
a, like a little sip of water, for example?
Well, the cells like that we use
Yeah.
when we culture them, we usually store them in 1 mil of their media.
Okay.
So we usually get like about 500,000 to 1 million
wow,
cells. That's not too much, but that's what you usually use.
That's
So
$1,000 for the
I think it's
500,000 to a
more.
million.
I think it's probably more for that.
Wow. Okay.
Yeah, but it's not. Yeah.
What's like a milliliter, just like so we can like visualize it? Like what would it look like if you saw it?
Like, have you ever used the 1,000 microliter pipette and have taken up like
I mean,
the PBS?
I
A water
did, but-
bottle, a plastic water bottle is 500 milliliters. So 1/500th of
1/500 of like a normal
plastic water
plastic,
bottle.
like a Nestle, like
Yeah,
plastic
it's
one.
not. It's like
So I could fill like a cap, like the cap of the water, for example, something
Maybe.
like that.
Maybe a little more.
My bad. I'm just trying to get like a
1
idea.
mil. It's less than the cap.
Maybe even a
Oh,
bit
really?
less, right?
It's
Yeah,
a bit less
wow,
than the
okay.
cap.
So
Yeah,
that
the cap's
shows
probably,
really
like,
that it's
3.
super expensive.
But then when you get that amount,
Yeah.
do you like then make it keep like
growing.
We haven't like gotten bone marrow from companies before. Like we usually get it from the donors, so we wouldn't really know how much we would get specifically.
Okay.
Oh, fair enough.
Yeah.
Okay.
Yeah. Right.
And I also wanted to rewind just for 1 second. Because remember you were saying, so you started your thesis and then went into your master's. So did you start that lab only for your thesis, and then through that you discovered like you want to continue in this lab, or did you do it before that, the same lab? Just for clarification.
Yeah, so I started in the lab that I'm currently at the beginning of my 4th year. And during my thesis is when I really found out that I like research and I like finding out things and being the one to design my own experiments and carry them out. And so that's when I decided to pursue a master's. And my PI offered a couple experiments, so either to continue the one that I was working on in my undergrad or to start a new one. And I thought that starting a new one would be really cool. And so that's just what I did.
All right. So what do you guys hope to achieve after completing your master's?
So for me, my future plans look like hopefully pursuing a PhD after my master's, or at Western, you have the option to transfer into a PhD program after 5 terms being in your master's. So, for example, like around this time next year, I could request from my PI that I want, or like bring it up that I want to transition into the PhD program at the same lab. And if he agrees, then I could also talk to my advisory committee, which is a committee of other PIs that kind of track my progress in my degree, and if they agree, then I transition into the PhD program. So that is my goal for like currently. That is what I'm working towards. And and then that would look like, so in your master's you work on 1 project, in a PhD program you work on 3 projects.
I didn't know that. Okay.
Yeah, so it's 3 to 4 years and you have 3 projects to work on.
I see.
Yeah, so
it's
very
not
excited
just
about
one. It's not like one project for like a continuous amount of time. It's like or like for that 3 to 4 years, it's like a bunch of projects. And
yeah, so it will still probably be about the same topic. So I'd probably still be working on stem cells and my mouse model and like diabetes. It will just be like a continuation of what I'm doing right now, or seeing where it takes me.
And,
Yeah.
Is it 3 projects simultaneously, or like 3 projects just in total? Like you finish one, then go to the next?
We'll see how it goes.
Yeah?
Like I think simultaneously.
And a lot of the times, it's not very clear cut when one ends and when one finishes.
Exactly.
They kind of
They
roll
overlap.
into each other.
Yeah.
They overlap. And also for the 3 to 4 years, I just want to clarify, is that after the 1 to 2 years of your master's, or is that PhD alone? Just because, a lot of people who are considering rolling over, they might not be sure about the timeline.
So it's 3 to 4 years after your first year being a master's student. So total time is 5 years on average.
Oh, okay.
Yes.
Oh wow, that's a pretty long time then, right?
Yes.
Yeah, okay. That's interesting. So it's like I was thinking that like the PhD would be a very kind of like similar to like your undergraduate degree, where it's like you take these X courses and you do this stuff. So but it seems very like kind of unordered in a sense, right?
Yeah, so grad school, like master's and PhD, are very different from undergrad. That's something I've realized. It is nothing like undergrad. We only have had 1 course, and once we're done that course, we don't have any courses anymore, ever.
Okay.
And that course is not your traditional undergrad lecture type courses. It is a course on how to be a grad student.
Okay.
So the main things that we do is focus on how to present her findings, how to give a good presentation, how to
Develop science communication skills, basically, and writing skills. So we're currently working on writing a grant of our proposed research. So yeah, it mainly focuses on grad level writing and communication skills.
I see. So in this course that you're taking, that's like making you a better grad student. You mean like they're teaching you how to write grants and apply for money and stuff like that, right?
Yes, exactly.
Okay, cool.
And then after you're done the 1 course, is it more just like you're working in the lab doing research? So what would you find is the difference between doing a master's or PhD versus just working as like a research like assistant? If you know.
So they are similar in a lot of ways. I guess if you're just working as a like a lab manager or a lab technician, you're not working towards a degree. You're just, that's your full-time job.
I see.
While as like as a PhD student, you're working towards your PhD degree or your master's degree, I guess.
I think in a lot of big labs as well, the purpose of a research technician is to also support other projects going on.
Sure.
So as a grad student, you have a project that you're working on with a definitive goal. But just in our lab, for example, our lab manager, she doesn't have one specific project. She helps everyone.
Yes.
And so her role is
And we can't do anything without her. She's the greatest.
We can't do anything without her. No, she's the best.
And she trains everyone too. Or most people.
So especially in our lab right now, we have a lot of master's students. So there's, I don't want to say a lot of turnover, but compared to a PhD or postdoc who's in a lab for 5-6 years, a master's student is there for 2 years. So there's a lot of training that must be involved, which is what our lab manager does. And another difference, I think, would also be what you're working towards. So a lot of people who are in grad school, especially if you want to pursue a PhD in academia, they want to become a PI. And so in order to become a PI, you need a certain number of publications, and you need certain
postdocs.
Yes, and you need postdoc as well, which is kind of a training period that you do after PhD. If you
kind
would
of like residency from med school. A little, you could think about it like that.
Right,
Yeah.
but for research. So there's
postdoc, right?
Yeah.
Okay.
Postdoctoral degree.
Okay.
Yeah, so there's quite a bit of differences, but I think those would be the main ones.
Yeah, I think we covered it mostly.
Okay, sounds good. So maybe we'll move on to the next question, or how you guys? Okay, sounds good. Okay, so, could you guys tell us what a typical day in your life as a master's student in regenerative medicine would look like, or like what you guys would be doing and stuff?
So for me personally, my day, it does not look the same every day. So for some labs, some master students might have a more 9-to-5 type schedule. But for me, because I'm working with cells and Nouran's working with rodents, so with mice, we don't exactly have a consistent schedule. It really depends on what my cells or her cells or, the animals need. And so typically we might be there some weekends, we might stay late some evenings, we go early sometimes, but there is quite a bit of a work-life balance, especially because we have an understanding PI, which I think is really important. But generally, I would say we put in about 40 hours a week, kind of like a full-time job.
But they divide like unevenly, we'll say. Like on some days we'll stay longer, some days we will leave earlier. Like today we left early to come here and talk to you guys. But other days we'll stay until 6, or some weekends we would come in both days, So I think on average it does add up to 40 hours a week, but it's not consistent on like each day.
Yeah. Right, and it also weekly is not consistent. Some weeks will be there a lot longer, especially if we have a lot going on.
We have a staining protocol.
Yeah, but I think ultimately it comes down to your planning as well. Yeah. I mean, there's only so much you can plan, things happen, but I try to plan it out just so I do have a work-life balance.
Yes. Thank you, Yina. And then I guess, wait, sorry, did you want to ask a question? Okay. So a day in my life as a graduate student. So for example, today, it's a Tuesday. So in the morning we have our weekly lab meeting where everyone in the lab, like all the students, we meet and we share our like data, what we did last week, what our plans look like for the following week. And we basically catch up our PI and our lab manager and all the students in the lab on what we're doing. And then after that, I went down to do some animal work, which Yina helped me with a little bit. And then I came up and I did some work. I did some data analysis on my computer, and then we came here. So that's what my day looked like today.
I see.
So that's pretty surprising then that like the day isn't like very, very structured. Maybe like for some uni students, I'm being honest. Like I'm not a very good uni student. Like sometimes I miss my lectures and stuff. I do them like online. But like for a typical uni person, they're going, like every day they have a kind of a schedule to their lectures and kind of an order. So there's kind of that like.
There's definitely an order of. It depends on how I plan my week and how my cells are doing or how my animals are doing and what they need from me that week. So, for example, sometimes I have surgeries or treatment days that week. So my week would look different from the following week where I don't have surgeries or treatment. So it's definitely how I plan out my week. I do go to the lab every day from basically 8 a.m. to about 3 p.m. or 4 p.m. It just every day looks a little different.
Could you be more specific about what you mean surgeries and treatments?
I knew that was coming up. So part of my study, or even Yina's as well, so remember those secreted factors that I was talking about that we get from the multipotent stromal cells, the cocktail.
Is that a term?
No, it's actually a term that Yina specifically uses, not me.
Okay.
So I just call them MSC secreted factors.
Come on.
It refers to something else, so we inject the secreted factors into the mice, and we inject it directly into their pancreas, so that requires a surgery.
Okay.
So we an incision, and then we inject the conditioned media or the secreted factors into their pancreas, and we suture them back up, and then the weeks I do have surgeries, I have to monitor them for the following week. So every day I have to go down and check on them, make sure they're okay. That's what it entails.
I got a kind of a random question. Like when you work with mice and stuff, like do you kind of like build like a relationship with the mice, like it's like a dog or something? You know what I'm saying?
So for me personally, I was not always the most comfortable working with animals. So it's still honestly taking me time to get used to them. I think they're super cute. I love them, but I haven't given them names or anything like that. Like I know my friends sometimes go like, "Oh, what'd you name them?" I'm like, I can't do that because I'm not super comfortable, doing that. But they are super cute. I love them. They scare me sometimes, but for the most part, they're super cute.
And what about? Are you working with animals? Sorry, or?
So I have worked with animals on my previous project. Right now, I don't have any going on. I go down with Nouran sometimes. We work with the mice, but right now I don't have any mice of my own.
But they are coming up for you.
They are coming up.
Yeah.
And I was also just wondering, what does the application process look like for a master's? And also, do you guys get any breaks? Because I've heard that master students don't have reading week or summer break. Is that true?
Unfortunately, that is true.
Oh, no way. Okay.
So, our program is 24 months straight in length.
Oh, wow.
However, we do get vacation days.
Okay.
So,
days,
aside...
not even weeks.
So, aside from, statutory holidays, we get, I think, about 2 or 3 weeks of vacation. I'm not exactly sure what it is.
It's pretty good, to be honest.
Which is
if
pretty much like a full-time job.
Supportive and understanding. If you ask them for a day off because you had to go home and do something, then he'll ours says yes. So it depends on many factors, but
Yeah, so we do have vacation days. We do get breaks when we need them, when we want them. So.
Okay.
In terms of the application, when are those usually due? And is there essays you need to write and an interview? Is there that whole process, or how does it work?
Yeah, so I was actually very confused about that when I applied last year. As for the deadline, I think it varies between programs.
And it's a rolling deadline.
Yeah, and it's a rolling deadline. So, do you want to explain that? I'm not really sure.
I think they have multiple dates where they send out acceptances. So the earlier that you apply, you find a PI who will take you, then you can get accepted earlier. But if you wait a little bit longer, if the seats fill up in the program, then they might not be able to accommodate you. But if there's still space in the program and you apply later on, then and you are successful, then
Yeah.
yeah.
And I think it also depends on, like, when Yina says there's seats in the program, like, how many students is a PI willing to take into their lab in the upcoming year. So sometimes, like, for example, when we applied, our PI wasn't taking 2 students. That's how we both got into the lab. But sometimes he can only take 1 student. So that differs between programs and labs. And so the traditional way of doing it is you apply to the program, then you get accepted, and you write like a letter of interest, so like expressing why you want to pursue graduate school, and you also have to submit your transcript with your grades. And then once you get accepted into that, you look for a supervisor who's part of that faculty that or department that you applied for. However, like in my case, I interviewed with the PI first. Then I applied to the program. So it's like there's kind of multiple ways of doing it. But I think it's more important to have a PI that you know will be able to take you into their lab than like just the traditional application process. Yeah.
Okay. Wow, that's actually really insightful. I did not know like how this all worked.
Yeah. I didn't know either.
It does vary between
Yeah.
departments, so this is how it works for the physiology and pharmacology department. Can't speak for anything else.
Yeah.
Okay.
Good point.
And when are applications usually, like when would you recommend to start applying or start preparing your application?
Probably as early as possible,
Yeah.
but I did not apply till I think midsummer, so maybe July, I want
Yeah.
to say.
I applied at the like mid-March, so like this time last year. I applied, but that was after I interviewed with my PI.
I see.
Yes, and I honestly thought I was pretty late to apply because I was talking to other students that interviewed with multiple labs, like, and they had been interviewing since like December or January of that year. But it's very variable. It really depends on the PIs you interview for and talk to, and the department or the specific program that you're applying for.
Yeah, I also want to add that one thing that kind of sucks, I would say, about Ontario is that it conflicts a lot with the professional school rollout.
Yes.
So I know that medical school results don't come out till mid-May. And that is really stressful for a lot of people because if you don't get in, you don't know what route you're going for. So I had applied to medical school last year and I was waiting for my interview results, and it wasn't until mid-May that I found out that I wasn't going to get in. But I had already talked to my supervisor and I already planned out before that I would be doing a master's. But I know that for a lot of people who are still waiting, it can be really stressful not knowing
Yes.
what you will do next.
Yes.
And so what I would recommend is, having that alternative route planned out and just having something that you can pursue
If that's your plan.
if that's your plan, yeah.
I just had one question related to that. Like, so, because you guys are in masters, and obviously there are probably other people that have just finished uni that are with you, and most certainly there are other people that are with you that are maybe wanting to apply to medicine after they finish. And then some people, for example, like Nouran, wants to continue in research. So in terms of like, I would say for people that would want to, for example, continue in medicine, is there a very high benefit of after university going into a master's? Or like, for example, there are some people after they finish the undergrad, they work or do something else other than a master's, not academia related, maybe try to buff up their CV.
So I would say to not pursue a master's just to fill in that time. I would say you have to be really interested in the project and you have to be willing to do what it takes to go through with it. Because, like we talked about before, depending on your project, masters can look very differently. And so I think it's a great way it's a great thing to do if that's what you enjoy doing and if it's what you can see yourself doing. So I was lucky enough to have that experience in 4th year, so I kind of knew what I was signing up for, and I really enjoyed my project and I enjoyed working in the lab that I was in. I do have friends that within this year, they instead of pursuing a master's, they have been working as, for example, just a tech or just working in a lab as a student. And I have other friends who have pursued masters but not thesis-based, so course-based masters.
Okay.
So these are traditionally one-year masters that instead of writing a thesis, you take traditional lecture-based courses, you write exams. So there's differences in this as well. For me, I wanted the experience of taking the 2 years thesis-based masters, and I wanted to do the work that I had started. So,
and you enjoy the project.
Yeah.
And what it entails. Yeah.
So for you, in your circumstance, it was kind of almost a continuation of what you were doing in your last year, which was why it made sense to continue in the thesis route. To continue in like the master thesis-based route, correct? Or
yeah, I wasn't really interested in doing a course-based master's, just because it it just wasn't really something that I considered. I looked into it briefly in my third year. There was this really cool microbiology and immunology course-based masters that I wanted to do. The difference is for course-based masters, you have to pay out of pocket. It's about $20,000 for tuition for the year.
wow. That's
Whereas,
very
as for a master's students for thesis-based, we get paid.
yeah, it's very
It's
interesting.
it's
We still pay tuition, but we do get paid.
yeah.
Yes.
So we have a stipend,
Yes.
which
What does that
helps a
term
bit.
mean? Stipend?
So stipend just means the funding that we get as a thesis-based master's student, and it varies between your department as well as your program. So I'm not sure what it looks like across the board, across Schulich or even across Western, but that is also one reason why I decided to do the thesis-based masters.
So for any of the money makers out there, what is this difference in the tuition versus the pay on average, right?
Yeah.
Maybe, yeah.
Not a lot
Not a lot.
at all.
Okay.
We also, in our program, like in physiology and pharmacology, we are guaranteed to have TAing experience.
Okay.
So that is not the same across other departments. So we do also get paid from that, but it's still.
But our TA money is included in our stipend. So if you
Yeah.
don't TA, you lose part of your stipend.
Okay.
Whereas for some other departments, when you TA, you get additional money on top of
But
your
it's not
stipend.
guaranteed.
Yeah, but it's not guaranteed. And as for the difference, it's about $10,000 between what we get and what tuition is, I think. So after you take out rent,
That's a big bag. That's like a
rent, groceries,
that's like a nice
you know.
trip. That's a nice trip.
Well, it's a nice trip if you don't eat for a year.
Fair enough. When you take all those into account.
Yeah, for real.
Sorry, I'm just a little confused. When do the applications open? Because I kind of hear people are applying all throughout the year, or it depends on the specific school, or
So I think it depends on the specific school and the specific program you're applying to.
okay,
Yes.
okay, makes sense now.
I think it opened up in January or February, but again, it is a rolling basis. So I think you can apply up until like end of August or something.
Yeah, technically.
But it depends.
And also, like for masters, like thesis based, or as far as I know, in our department, you could start in May, in September, or in January. So you could start your masters at different time points. So you don't have to start in September like undergrads. Or I guess undergrads sometimes they start in January too, in some circumstances. But yeah, so I guess that depends on when you apply as well, but there's definitely that option. If like you miss the September deadline, you could definitely start in January if that's what you and your PI agreed on. Yeah.
Right. Or if you want to take a semester off between undergrad or a year off between undergrad and masters, it's very flexible.
Yes. It's just open communication with your PI, I would say, is the most important thing with that.
I just wanted to ask one more question. So, like, not to sound very blunt or anything, would you say that, like, because you're talking about, I think, like, that's a big thing, especially from what I'm seeing around with my colleagues and people in my upper years, like, what are we going to do after we finish fourth year? And then you guys are discussing that there's kind of this variability. Some people go into masters, some people work. To say bluntly, do you think that there is kind of a larger effort or kind of more mental strain, mental effort, kind of more focus needed to pursue a master's than, for example, someone that does a degree? Because then that might influence someone's decision on, like, you know, should I pursue that because it's just going to be way more effort, or should I do another thing if it's not going to be as, like, beneficial? Do you get what I'm saying? Like, kind of?
So as far as from what I understand, pursuing a master's definitely takes it's continuous learning. That's how I view it. And I am an advocate for that personally. I think that is my purpose in life, to continue learning, and that's why I want to pursue a PhD also. So it really depends on what you like, what your values are in terms of academia and how much you want to learn. I know there are some people who look at me and they're like, how are you still in school? Like, like you're done undergrad, like get out of there, get a job. And I'm like, I can't imagine that for myself. I can't imagine myself not being a grad student currently. So it really depends, like, on like person to person. I can't say like it takes more of a mental effort to be a grad student than like someone who would choose to work, because I'm sure like work also requires a lot of like mental effort, but it's different. It depends on you and what you like.
And I think it also depends on your end goal.
Yes.
So, for example, like Nouran, because she wants to pursue research, this is a stepping stone that you kind of have to do.
Yes.
So,
yeah. For sure.
I see. So that's pretty good, because I would say for a lot of people in my position, by the way, like I am an undergraduate student currently in my second year, and people obviously in upper years, I think a very common misconception is that you must continue in like a master's before you would either want to, I don't know, even people that want to work in like industry, or like go to medicine, you know. But it seems that you're not, it seems that isn't the case. That's what I've learned from what you guys have said.
it is possible to get research positions right out of undergrad, especially if you have research experience in your undergrad. But in terms
there isn't much growth.
Right. In terms of
once you're
growth
employed.
potential, it really does help if you have an extra degree, whether it be a master's. And of course, the more degrees you have, the more growth.
Yes. And I would say currently, like on the job market, I was looking for like industry level positions. Like when I finished my undergrad, I just wanted to see what's out there, and like I was looking at like medical writing jobs or medical counseling jobs in like pharmaceutical companies or research companies. Most of them do require a master's, is what I found through my search, or a PhD, but definitely a master's. They all require a master's. So again, yeah, like as Yina said, like depends on what you want to do, on your end goal. I think that's how you should start planning out your next steps.
Okay. That's good though, that there's kind of like, it just really depends on what you want to do and what your interests are, and you know, so that's a good thing. Anyway, so for next question, which is slightly unrelated but it's a pretty interesting question. Have you encountered any significant challenges during your academic journey, and how did you overcome them?
So for me, when I came into undergrad, I was under the impression that I wanted to pursue medicine, that I wanted to become a physician. And through first year and second year, I was like, yeah, sure, like, I guess this is something I want to do. But the more I learned, the more I realized that I did not want to pursue medicine. So at one point, I think it was in third year, I was like, okay, so what am I going to do? I don't know what I'm doing, because I hadn't been exposed to research at that point. And it was a pretty low point where my parents thought I was going to med school, you know? I thought I was going to med school. I did not even want to write the MCAT because I knew I didn't want to do med school. So I didn't know what I was doing. I considered other options. I've always wanted to become a teacher before. Like, teaching is something that I've always been passionate about. I've always been a tutor for my cousins, for my siblings. I've worked in like teacher shadowing before. So I considered that, but there's a lot of obstacles also with becoming a teacher, and like, you know, like being a little bit more well off later on in life. So I didn't end up going with that. So I was really struggling to find my purpose. And that kind of resolved when I was in the lab course in my lab course in IMS in fourth year, where I was exposed to research more. And I was like, wow, this is very, very cool. And that's when I decided to pursue research. But yeah, before that, I was kind of lost.
I think for me, the challenge was my own misconception of what research was. So I had a bit of exposure to research throughout the early years of my undergrad and before, I'd visited labs. But what I would do when I went was just refill pipette tips, and I would do really mundane tasks. And I remember thinking, during my second and third year, I am not going to grad school. I am not going into research. This is not something that I wanted to do. And it wasn't until I finished third year, and I think third year is when I took most of the courses that gave me the knowledge that was required to be able to understand what a lot of labs are doing. And during my volunteer experience that I talked about before, that was when I started really understanding what was going on in the lab, what I was doing and what other people were doing and what my findings presented. And I think that was exacerbated a lot in my fourth year when I had my own project and just being able to work on something yourself. And when you have a finding that you are the only person who currently knows what is going on, that's like a feeling that is very different than anything. It's novel, exactly. So I think for me, the challenge was just overcoming that stigma that I had towards research.
Yes.
And, just understand that your mind can change. And I think that's the big thing for both of us.
Yeah. And just to add on to that, what Yina said about her misconception within research or what research was about, I think in our early years of undergrad, like especially like me and Yina were talking about this before, but in our first year labs or second year labs, like I think this is like a unanimous experience where we all hate labs. Yeah. And I didn't realize it until honestly starting my master's that is not what lab is. Like what the research you do in your master's is definitely not like the research that we were shown in our first or second year lab experience. It's just,
no hate to the coordinators.
Yes, no hate, of course. But it's very different. I think it's like they do as much as they can. It's like if you don't have a set project, you can't really be exposed to it too much anyway. Mm-hmm. So I don't think it's their fault per se.
No. And as a first year and a second year,
you don't even know really what's
going on.
I didn't know
what was going on.
Yeah. So don't, like, if you hate first and second year labs, don't just not consider research because you think it's going to be like that. It won't be like that.
I think that's, thank you guys for sharing those experiences. I think that's a really, really good perspective that you have. I think a lot of people are, for example, for Nouran, are in the same boat that you were in, that thinking that, especially going into a degree like med sci or health sci, anything science related, people are like, I want to do medicine. So that's good though that you kept an open mind and you were able to change your decision, right? Regardless of course, for everybody, the common experience is that there are outside factors that influence, your decision. Like it's not only what you want to do, it's maybe your parents, your family, what your friends think about you. So that's good that you had the courage to again have open mind and change that.
It definitely took a lot.
Yeah. And for Yina, that's a good, also your perspective, I think, is something that I would need to think more of actually, because to be honest, in my second year of undergrad, I am not, or I was before listening, I guess, not super open to research. So I think that it's good that you were explaining that you had that experience where you weren't open, but then you realize that, that don't just believe any stigma or what people are saying, you have to understand it for what it is, and then you can make your decision accordingly. So yeah, that's great, guys. Thank you. One other question regarding that. So of course, we're looking a lot about the master's life or master students' life. Sorry, the work you're doing, the time, all of the factors related to it. But outside of the lab, what does it mean also to be a master student? Other than conducting experiments and things like that. So yeah.
Yeah. So that's a great question. So outside of the lab, there's a lot of scientific outreach opportunities and science communication opportunities that master's students take a part of. And one of the biggest examples is going to conferences. So going to conferences basically means that you go to a big event where it's usually about, it's themed. So for example, the one we usually go to is a stem cell themed conference where different researchers and their students are invited to present their research at said conference. So you would have your poster where you present all your data and your research, and you take it there and you present it to people who, like your colleagues, other PIs, and that opens up room for networking. It's a huge event for networking and collaboration opportunities. So I know where Yina gets her cells from Toronto, that was due to a collaboration between our PI and the PI of the other lab in Toronto. So it's definitely a great opportunity if you can go to conferences as a master's student. It really builds your communication skills and your networking.
Perfect. Thank you so much for that response. Okay, so Laura, take it away.
And if you could go back and give advice to your pre-master self, what would it be?
I think one of the main things that I've learned is that expect the unexpected. So especially when we work with cells or animals, things that are not predictable, there will always be something that goes wrong. And so I would just tell myself that things will not go to plan, but that's okay because everyone experiences this, and we're lucky to be in a supportive and collaborative environment where, everyone is very understanding and very willing to help each other through these. Yeah.
Things will go wrong in your masters, in your protocols, but that's okay. That's what research is about. It's trial and error.
And life also.
Yeah, with life too. I guess that applies. Yeah.
And I'd like to add that that's how a lot of cool scientific discoveries have been made.
Yes.
Through mistakes. And
the animal model I use was actually discovered through a mistake. So
look at that. And look where it's led.
Yeah.
That's pretty cool. So finally, we just wanted to touch one more thing, which is, what would you consider the most rewarding aspect so far pursuing the master's degree in regenerative medicine? And take it away.
I guess having a lot of knowledge about something so specific. It feels really nice to, all the readings I do or we do in the lab are just about diabetes and stem cells, but obviously we do incorporate an interdisciplinary approach, but you just get so caught up in all the details about your project and about the field that what you're doing, that it feels like, you're the only person who knows this, which is very cool. I love that feeling. Or as Yina said, when you make a finding, no matter how small it is, you're the only person sitting on that information currently in the world. So it's very cool. Yeah.
And just to conclude, looking toward the future and stuff, are there any specific goals or projects, in your further education in master's or PhD in your postdoc that you want to accomplish, something that you have in your vision and you see, I need to figure this out, that problem?
Well, I guess one exciting project that is on the trajectory in our lab is a combination of my project and Yina's project. So combining regenerative and replacement therapies for beta islets in a mouse model that has autoimmune diabetes. So I use a model that has autoimmune diabetes. Yina will use a model that we induce the diabetes in, or induce hyperglycemia. And I shouldn't say diabetes. But combining both will open avenues for more clinical applications in the future too. So that would be pretty exciting.
Yeah, I was going to say something similar. I don't have a specific project in the future, but I would really love to see the work that I am doing now having an impact on clinical translation, or seeing, things that I'm working on be translated into modern medicine.
Yeah. That's definitely something I can understand, because it's for me, for example, one of the reasons why maybe I wasn't too interested in research is that lots of people tell themselves I don't think I have it in me. I don't have that kind of light or that ability to discover something which changes, the face of the world.
Well, discoveries don't happen overnight, right? It's the small things that you put in, the small steps in your protocol that changes a small thing in your results that can lead to a big discovery. But the work you've done is also led by work that has been like has been going on for like 30 years or 40 years or 50 years, you know? So discoveries aren't made overnight. There's always like background.
Yeah, there's research that's been going on for a really long time that's still not wrapped up, and I don't think we can definitively say that anything is ever completed. Like, for example, in our lab, we've had multiple students work on the same project, and that's completely normal within the field. For something to take years to develop and for a lot of people to be working on the same project. So
that's a good thing because it means your results are actually valid if there's multiple people working on the same project and getting similar results.
Right. So I would say don't put a lot of pressure on yourself that you have to make a discovery or you have to find something new.
Yeah.
That's
My
great.
expectation of myself is not to cure diabetes. I mean, hopefully, but I don't expect that of myself.
Right. Exactly. So it's the small things, like Nouran said, even the really small finding, even if it's a negative finding. So even if it goes against your hypothesis, now you know for sure that that's still a result.
You could take a different route.
Exactly. So just because it goes against what you thought might happen, or just because you find the opposite result, doesn't mean that you don't have a finding.
Yeah. And actually that's good now. So I understand. I think that's a really good message to bring, because it shows that a contribution doesn't only mean discovering something new. It could be like maybe proving something wrong that helps everybody else that's going to do research in it. And also that you're saying it's like it's a continuous process. So maybe for me, like my characters, I'm kind of very impatient. Like I want to see results right away. So maybe for me, like it's that's why my mind goes to that. But I understand.
Motivate you to work harder.
Yeah, exactly. It can motivate you to work harder. And I also thought, but still though, regardless, like I think that's really good that back to like what we were saying before, that like you have this desire to see a clinical application for your research, right? Because even, you know, like I 100% agree now, like the contribution can be made different ways. Like there's something different about, like, for example, you go in a store and you see that your treatment or like you see in the hospital, you see your treatment is being used to help people make their lives better and things like that. So I think that's really beautiful.
Yeah, I definitely find that it's probably very rewarding knowing that the work you're putting in has the potential for something so great. So yeah, I really appreciate you guys joining us today. Thank you so much. I found this super insightful.
Thank you so much, guys. Thank you, Yina and Nouran. Thank you for coming. You guys have been excellent guests. We are so happy to learn from you. We've learned a lot, and hopefully our listeners as well will learn from their very deep insights and good experiences. So yeah, thank you so much, guys. We really appreciate it.
Thank you guys for having us, and we just like to thank all of our lab members, as well as our PI and my collaborator, Dr. Nostro, just for being able to support us and making all of this possible.
And if you want to learn more about our research and the work we do in our lab, feel free to visit our lab website, www.theheslab.com, which me and Yina created.
Check it out right now.
All right, thank you for listening to the CARRM's Profile podcast. Until next time.
Bye.